All right, so this is your EOC cells review. As you go through this is not meant to be something that you take notes on. It's meant to be something that you just kind of watch and look at the diagrams and read things through. There may be certain points where it would be in your best interest to pause and then really examine the diagram, but otherwise just kind of listen and follow. So first up is the phospholipid bilayer, which is of cells. Hopefully you realize that. And it makes the cell membrane, but it has two parts to it. So there's the head part and the tail part. Now there's some key words here: hydrophilic, hydrophobic. Phobia is fear. Philic is love, and then hydro is water. Okay, so the heads face out because there's water here and water here, and the tails face in because water doesn't go in the middle. So they're water fearing, and they don't want to touch the water. The cell membrane is also sometimes termed the fluid mosaic model, and this is because many molecules float in the membrane, as you can see from this picture. These molecules include proteins. There's 5 different types, but there's only a couple major ones that you need to know: cholesterol, and then carbohydrates. So protein functions. So proteins have 3 major functions. So they transport signals if they're a glycoprotein, which is this one right here. They support structure if they're an integral protein. So this is an integral protein, this is an integral protein. And then they transport things in and out. So these are your channel proteins. So things can go out, things can go in. The cholesterol just prevents those tails from sticking together. So they're kind of randomly in between the tails, and they prevent it from getting stuck. Carbohydrates they identify signals. So they exist in 2 ways. They're either on a protein or they're independent on one of the heads. So types of transport through the membrane. So the first thing is passive transport. This is no energy needed. There's 3 different types: diffusion, facilitated diffusion, and osmosis. So diffusion, really important, is the movement of particles. And because it's passive, it's always from high to low, always, always. And diffusion moves things like lipids, oxygen, carbon dioxide. If you kind of look over here, you can see that moving through. Osmosis is simply the diffusion of water. The way I remember osmosis is H2O, that O, that starts with an O. That's how I remember osmosis is water. And it's water only. That's it. All it moves is water. And once again, from high to low. Osmosis can also result in 3 different things. So in a hypotonic solution, a cell is going to swell and it could burst. But if it's a plant cell, it won't burst because of the cell wall. So the cell wall prevents that cell from bursting. And actually, plants love this. This is what plants want to be, is hypotonic. Isotonic is normal, equal. This is moving in and out, as you can see from the water arrows. And this is a normal cell down here. This is like blood cells. Whoops. Blood cells look like this. So blood cells and most of our body cells, they like to be isotonic. And then hypertonic really isn't good for either plants or animals. This is when it shrivels up. So it shrinks. If you look down here, you can see the cytoplasm pulling away from the cell wall, all those spaces in between. That's not good. Active transport, then, is the movement of particles across the membrane using energy. This is going from low to high concentration, which is against the gradient, uphill. So there's 3 types here. So transport proteins, endocytosis, which means in, exocytosis, which means out of the cell. So prokaryote characteristics. So we talked about 2 types of cells, prokaryotes and eukaryotes. So prokaryotes are the simple ones. They're simple, they're single-celled, they don't have a true nucleus, but they do have DNA. A lot of you forget that. You think, oh, no nucleus, no DNA. That's not true. They still have DNA. And they have very few organelles. There's 2 different types. There's archaebacteria and eubacteria. So eukaryotes versus prokaryotes. So this is kind of just comparing the two. If you look, prokaryotes, like we said, are small. Eukaryotes are large, not as complex, very complex. These have that nucleus. These no nucleus, but they do have that DNA. Don't forget that. Chromosomes, single strand of DNA, membrane-bound organelles, no membrane-bound organelles, and then examples, animals, plants, protists, and fungi. These are just bacteria. So prokaryote structure. So there's 5 major portions we learned. There's a few more in the picture. DNA is in the middle. Flagella is your tail. Pili, those are all those little hairs. Okay, and then ribosomes, all the dots in the middle. And then the capsule is that outside surrounding. Now, if you were asked any other parts of the structure, you need to know that there's also a plasma membrane or cell membrane. And there's a cell wall. Okay, so the capsule, most of you recognize, but below the capsule is a cell wall, then a cell membrane, just like, kind of like a plant cell. So this is a eukaryotic cell. Don't forget that. So this is an animal cell. You should be able to identify major things and what they do. So you have like your Golgi, your rough ER, smooth ER, mitochondria, ribosomes, cell membrane. Those are the basics. Oh, and don't forget the nucleus. Plant cell has a few different things. So the vacuole, don't forget that. Golgi is here. This is your chloroplast. And then mitochondria are a little bit smaller. Okay. And then you have your nucleus and your ER. Those are the major things. Oh, and don't forget about the cell wall. So on to photosynthesis. So there's 2 major parts of photosynthesis. There's light dependent and light independent. You know what these words mean. So dependent means it depends on light. This is where the sun is used. Independent means it uses ATP because it doesn't depend on the sun. So a little about ATP. So ATP is short for adenine triphosphate. So adenine plus a ribose, which we know is a sugar, and then 3 phosphates. So it's an energy storing molecule. It's much more important in cell respiration than photosynthesis, but it's still important all the same. And it's the source of all energy for the cell. And what happens is one of those phosphate groups, or that P, breaks off, energy is released. So you get ADP, which D stands for diphosphate, plus a loose phosphate molecule. So if I were you, I would pause this right here and study this picture. The green are your in arrows, the red are your out arrows, and then the yellow arrows have some important information. So pause here and kind of study this picture. All right, a little review. So this formula, number 1, six CO2 plus 6 H2O yields 6 C6H12O6, which we know is glucose, plus 6 O2. What makes glucose and oxygen? Plants do, so that's photosynthesis. Blank is the process which breaks down food molecules to release energy and takes place in the mitochondria. Key hint there, respiration. Blank is formed when ATP releases a phosphate. We just talked about that ADP because it's diphosphate. Fermentation is a form of blank respiration which occurs without the presence of oxygen. That's anaerobic. And then the green pigment present in plant cells and allows them to go through photosynthesis, chlorophyll. So this is cell respiration overview, which once again I would pause here and examine this diagram. So an overview. Respiration, we release energy. That's what we want. We as humans do cell respiration, and we need that energy for our body to function. And we break down food when oxygen is present. Okay, so here's your formula. You should have that memorized by now. Fermentation is a type of anaerobic. Okay, so when you hear about anaerobic respiration, it's that. There's 2 types. Alcoholic, which we don't do. This is like bacteria that do this. And then our bodies do lactic acid. So this is like if you're doing like sprints or powerlifting, something that you're doing without regulating your breath, you're going to get a lactic acid buildup. The main takeaway point is one glucose molecule breaks down to between 36 and 38 ATP. So how are they related? They're pretty much opposite processes. So photosynthesis produces food. We take in food. And then photosynthesis uses carbon dioxide and releases O2. We take in O2 and release CO2. So cell division. So mitosis is cell division, which results in daughter cells from a parent cell. The really key phrase here is that the daughter cells are identical to the mother and daughter. This is like a clone, an exact clone. These are our body cells that do this. The stages. So interphase, mitosis, cytokinesis. Mitosis has 4 within it. Okay, so prophase, metaphase, anaphase, telophase. You need to have that order memorized, but you also need to be able to identify pictures at each stage. So plant versus animal cells, the big difference is cytokinesis. It's a cleavage furrow in plant cells. It's a cell plate, or cleavage furrow in animal cells, cell plate in plant cells. So here's just a picture. So as you see, they're lining up. You know that's metaphase. This is prophase. Anaphase, they pull together. Telophase, they're pulling even further apart. Cytokinesis, and interphase is over here. So uncontrolled cell division results in cancer. Cancer is just uncontrolled cell division, but of abnormal cells. And how does it kill? The abnormal cells crowd out the normal cells. So meiosis is different from mitosis. It's still cell division, but they're not identical. So the daughter cells have half the number of chromosomes, and eventually at the end you get 4 instead of 2. So really important as well, during meiosis, the DNA still only replicates once, even though the nucleus is dividing twice. Make sure you know that. So the first division of meiosis, we have prophase 1, metaphase 1, anaphase 1, telophase 1. You might want to pause here and read through each of these and maybe kind of do a little sketch in your mind of what it looks like. The second division, we have prophase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis is when your 4 haploid daughter cells are formed. Once again, pause here, go through this in your mind and kind of sketch it out or sketch it out on paper if that helps. So a little bit more about chromosomes. So what's a gamete? A gamete is a human sex cell. So it's either a sperm cell or an egg cell. Okay, there's 23 chromosomes per one. There's not 46 like your normal body cells. Couple key vocab words, haploid versus diploid. So N chromosomes is haploid. 2N or double is diploid. Di means 2. So differences between the two. Mitosis is asexual while meiosis is sexual reproduction. The cell divides once versus twice. 2 daughter cells versus 4. And then identical, different. Big, big, big point there. So karyotype. So you have these on your packet, but what does it tell us? They're grouped by size, but the other thing is it tells us gender. So males are XY, females are XX. Typically, on a karyotype, for XY, the X is really big and the Y is little. For XX, you're just going to see 2 big ones. And then also important to note, you had one of these on your packet as well. Make sure you are familiar with the disorders. So these are the 3 most common ones that you could be asked about. So Down syndrome is extra 21. Turner syndrome just has 1 X. And then Klinefelter has 2 X's and a Y. So why do we do meiosis? So it provides variation. That's why. Remember, we talked about this in terms of evolution. If everybody has the exact same genes for everything, one disease can wipe out the entire population. So we want to be a little bit different. So genes from 2 parents gives you that variety. And then crossing over can occur. And then the gametes randomly combine. So if there's 1 egg, there's going to be many, many sperm, but all the sperm are carrying a little bit different traits. So depending on which sperm fertilizes the egg will depend on what traits there are. So that makes it even more random. Asexual reproduction, it's a single parent and they're genetically identical. That's where we run into problems in terms of population and being successful. So a little bit more on eggs and sperm. So it results in either egg or sperm if you're an animal. It's pollen or ovules in plants. What's the difference? So female meiosis just makes 1 usable cell, whereas males makes 4. That's really the big difference that you need to know. Females usually stay in interphase until meiosis, which happens at puberty. Okay, and then fertilization is when an egg and sperm combine. They each have 23 chromosomes to make 46 total in a new human. So problems, we have non-disjunction, which is when they don't separate. That's this picture, if you look right here. So the X's should be separating, but oh look, this one doesn't have it. So if this is a sperm cell and it goes on to fertilize an egg, it's going to bring an extra chromosome. Other issues, Down syndrome, this is where we get 3. So if you look here, this one has 3, this one has 3. So if that's the egg or the sperm that's being fertilized, once again you have a problem because you have an extra chromosome. So that's going to result in that disorder. And that's it.